Good afternoon, everyone. This is Lydia from BMKCloud. I'm thrilled to welcome each one of you to our latest webinar. Today, we will explore visualizing key pathways and WGCNA analysis with real data. Our webinar today will last for about 30 minutes, followed by a dedicated 5 minutes for your questions and answers. You can freely submit your questions by using the Q&A button at the bottom of your screen. We are looking forward to receiving your inquiries, and if any questions are left unanswered, we will contact you to reply via email. Also, we'll share the slides and a recording of this webinar via email. In this session, we will explore advanced techniques for enhancing image presentations using volcano plots and heat maps to emphasize key pathways. Additionally, we will conduct a weighted gene co-expression network analysis, WGCNA, to identify important gene modules and interpret their biological significance using real data. We will cover the following topics in detail. Intersecting genes from the Venn diagram analysis. Learn how to obtain specific image, use target gene list, and save temporary results into BMKCloud in the case of Venn diagram. WGCNA and Results Interpretation. Perform weighted gene co-expression network analysis to identify important gene module and interpret their biological significance with traits. And the third, image enhancement and emphasizing target pathways. Learn how to enhance image presentations using volcano plots and heat maps, highlighting key pathways involved in differential gene expression. The session will help you master how to use real data on the BMKCloud to enhance image presentations and conduct in-depth gene co-expression network analysis, improving your research visualization and data interpretation skills. But before we start, let me introduce our speaker for today's webinar, Johnson. Johnson, equipped with extensive expertise in bioinformatics analysis, shines in his role. Holding a master's degree from Beijing Forestry University, China, he embarked on his professional journey as a bioinformatics analyst post-graduation, specializing in high-throughput sequencing data analysis, especially Pacific biosciences data. Johnson honed his skills with precision and dedication. His career trajectory led him to BMKCloud, where he transitioned to the overseas bioinformatics department. Here, he assumed the pivotal role of cloud platform product manager, steering the team towards innovation and efficiency. With a blend of technical prowess and strategic vision, Johnson continues to make significant contribution to the field, driving advancements in bioinformatics with unwavering determination. So let me hand over this virtual stage to our master, Johnson.
Thank you. Thank you. Hello, everyone. It's Johnson. Today, we will talk about visualizing key pathway and WGCNA with real data using BMKCloud. Long time no see. Let's review previous webinar first. First 3 webinars, we went through about main function in BMKCloud. We talked about how to submit analysis, how to download and update report, and how to visualize your results. You can customize your visualization results, filtering gene sites in multiple criteria, add group or delete sample using BMKCloud. And last webinar, we used published data to do a real transcript analysis with reference genome. Here are the material and our main goal. We did analysis of mRNA-seq reference with real data. First, let's see the material. All 9 seeds were grown on MS media in short day condition at 20 degrees for 21 days. Cold treatments were performed at 4 degrees in short days conditions for 3 days or 3 hours, and 3 seeds were treated as naive. So we have 3 groups. Each group has 3 repeats. And we have 2 trees as heads or flowers. I want to find how cold treatment influence and which genes plays main role. Here I set 3 DEG analysis compare groups, naive versus H3, naive versus D3, and H3 versus D3. Our main goal is to analyze the impact of cold stress on plants by examining gene expression. For this part, I think the whole analysis will follow these steps. Number 1, conduct analysis and check data quality. Number 2, review grouping conditions and assess whether analysis parameters need adjust. And number 3, summarize the current analysis result. And number 4, conduct advanced analysis and refine conclusion. And we have already finished the first 3 steps by far. So what we have obtained, we checked the first report and found that some samples are out of groups. So we update analysis procedure, remove some samples which are out of group, and get the final report. After updating final report, we've got target gene list for differential expression genes, or gene list of target functional annotation. We've got basic visualization results, including functional enrichment and gene expression cluster analysis. This webinar, we have 3 topics today. First, I will demonstrate to you how to get intersecting genes using Venn diagram analysis. We will get intersecting genes list and visualization result based on these genes. Next, I will introduce to you about WGCNA. This is a main additional analysis for transcript data analysis. The third part, I will show you about functional pathway enrichment analysis and how these results help us achieve our main goal. For the first part, I will demonstrate to you about Venn diagram. First, we will talk about how to get Venn diagram using BMKCloud. After we got the plot, I will show you how to save results by file, so we can find these results anytime you need. Then we can regard Venn diagram as a tool for us to get intersect gene list and do further analysis. So let's turn to BMKCloud, and I will show you step by step. Here you can see is the interface of the final report of mRNA-seq reference analysis report. Do you remember we just talked about we update the final report by removing some samples? Do you remember where we can find the update graph? Turn left to the navigation bar. Here you can see update the final report, new report. It may take some time, and you can see here you can find the updated report by remove the D3R1 and H3R1 samples. Click it. And so you can get into the final report we updated. You can check here. We only have the 7 samples because we have nine samples on the very beginning and remove 2. So where we can get the Venn diagram, you have 2 ways. You can use the whole genes, such as gene set Venn diagram, click it. And here are some parameters. You can filter out some genes by using the expression per level. Maybe you want to filter out some genes that has very low expression, maybe select to 1, or you can leave it by default. Here is the filtering method. You want to draw the Venn diagram using the intersection union or the mean by the samples. We combine the samples into groups, such as the group setting. I want this group setting same as the report. So I combine D3 repeat 2 and D3 repeat 3 samples into D3 group. The H3 group containing the samples of H3 repeat 2 and H3 repeat 3. For the naive group, I have the 3 naive samples. So after we select this, we can submit the analysis. This may take some time. This depends on how many genes do you have. Please wait a minute for this result. Oh, you can see here is the final report. This is the Venn diagram of the parameters we just set. On the top is the gene list of the table we use table of the genes we use for analysis, and on the bottom is the diagram. You can here see change the. Do you remember how to customize our visualization result? Here you can using the image wheel. Here are some navigation bar. You can change the text font family, font size. Maybe I want to make the font. I want to change the numbers larger. I can change the font size. Or I don't like this color. I don't want to use the MPG, so I can change the color of the Venn diagram. Or I want to hide the bar of the. So I can see here is the gram. Also, you can some other setting parameters. But here I want to tell you about another function. Maybe you have obtained this Venn diagram, but you have something else to do. So you have to break here. You can not only download the feature or the list to your computer, but also save the analysis result into the BMKCloud. Here, you can click save analysis results save. The BMKCloud will automatically choose the description of the analysis, and you can change the name of this analysis, such as today's data. September the 18th. So click OK. The result of Venn diagram you can find here, process record. This gene set when this is the result we just obtained. Gene set Venn diagram, September the 18th, and the description of the analysis. So you can here find the result you got anytime you need. Don't worry about the loss of the diagram. So we have 2 ways to get the Venn diagram. First, you can get the Venn diagram from the very beginning, select parameters, submit analysis, and got the result. Or you can find the result at the process record. And next, I want to tell you about how to do the gene filtering. Maybe we can regard this Venn diagram as a tool for us to get the intersect genes. Maybe I submit the analysis with 3 groups, and all there is the 15,000 genes of the intersect genes of 3 groups. I want to analyze specifically of these genes. So click the number. Click number. You can see data filtering button is clickable. So click it. Here is the very gene list for the intersect genes. The gene number is the same as the gene numbers in the picture. You have 2 further analysis ways. Number one, you can download the gene list for yourself and do the analysis. And you can use the BMKCloud analysis tools using retrieving the genes. Click it and you can see here is the same page of the gene search page, and you can do the further analysis or the one-click drawing. This is the same function as we talked before. Do the one-click drawing and these images will draw. BMKCloud will draw these images automatically, and you can use this gene list for the further analysis. So let's back to, this is the introduction of the Venn diagram, and let's back to our slides. Let's summarize here. We talked about how to submit diagrams step by step, using not only genes but all by DEGs. We can save and find previous results using BMKCloud, saving temporary results into process record. We can intersect gene using BMKCloud and using these genes for further analysis. Now let's talk about WGCNA. Maybe you've already heard of this method, but don't know details. Let me introduce to you simply. The full name of WGCNA is Weighted Gene Co-Expression Network Analysis. WGCNA is a technique that looks at the relationship between different genes in large data sets, typically genes expression data. The goal of WGCNA is to identify groups of genes that behave similarly across samples. These groups or modules may share common functions or be involved in the same biological process. So why we use WGCNA? The first reason is identify gene modules. WGCNA helps in finding cluster of genes that work together, which can provide insights into the biological functions of these gene groups. And reason 2 is link modules to traits. WGCNA can correlate modules with each traits to identify which genes networks are associated with specific conditions or phenotypes. And reason 3 is data reduction. By focusing on modules rather than individual genes, WGCNA simulates the analysis, making it easier to interpret larger-scale gene expression data. Here are the main steps of WGCNA, including 1, data preparation, number 2, network construction, number 3, module detecting, number 4, relating modules to traits, and number 5, module analysis. The in-depth understanding of this needs not only biological knowledge, but also mathematics background. For most of us, we only need to know that WGCNA helps us find many genes related to few genes, and find few genes related to many genes. Next, I will demonstrate to you about how to submit WGCNA using BMKCloud and go through the main results. Here is the main interface of the WGCNA submission. Let me introduce parameters of WGCNA in BMKCloud for you. First, we should choose about the genes we used for analysis. We can select DGs in specific compare groups. If you have many samples and groups, you can just merge samples, which will decrease complexity of analysis. Otherwise, you can use all your samples. Notice that WGCNA performs well using at least 5 samples or groups. Next part is gene filtering. You can filter genes using expression, such as filter out genes FPKM less than 1. For WGCNA module filtering, if you don't know how to choose these parameters, leave this by default. You can also add treaties in WGCNA, but notice that only quantity trees are acceptable. If you have quality trees, you should convert these trees into integers such as 0 and 1. Talk is cheap. I will demonstrate to you WGCNA using BMKCloud. Let's back to the BMKCloud interface, and you can simply find WGCNA here. You can do the WGCNA analysis using the whole gene or the specific compare group using the differential expression genes here. First, you must choose the genes you use. Maybe you can use all the differential expression genes all for the analysis. And group selection. If you don't have many samples, you can use all the samples for the further analysis, or you can combine. If you have many samples or the groups, you can merge the samples into groups for analysis. But remember that WGCNA performs well performed well, not less than 5 samples. So you need to have at least 5 samples for the analysis. Here is the analysis parameters for the module filtering. If you don't know how to select it, please leave it by default. And I want to show you about the treated section. Only quantitativity treaties are acceptable, which means you can use the treaties such as height. This is the quantity treats. Maybe the different symbol has different sets. You just type the treatise into the table, and the analysis will do the analysis automatically. Or you have the quality treatise such as the flower. So you must convert this trait into the integers such as 0 and 1. Maybe you can set the 0 as not to bloom the flower and the 1 for the flowering. After you set all the parameters, you can submit the analysis. This analysis will take a little bit long, and you can find the analysis process in the process record. Here you can see my WGCNA analysis submit earlier, and here is finished. If you just submit analysis, this tag will be the running. So let's back to my slide, and I will introduce you about the result of WGCNA. Let's go through about the main result of WGCNA. First table you should focus on is the gene list with module. WGCNA will divide genes into modules and use different colors respond for modules. Here in this table, we can find that the first gene, all of these genes are in the blue module. We should download this list or save this result in BMKCloud. This is very important. Next, we should focus on the result of module trait relationship. module trait relationship. In my result, this is my result. We can find that blue module significantly correlated with flower trait here. And the brown module significantly negatively correlated with head trait here. If we want to analyze further with these two traits, may this module need our more attention. I want to know how code treatment influence, so blue module and brown module fit my design well. We can find all gene modules relationship and functional enrichment results in download zip file. But the results here are for a specific module. I want to enrichment result of both two modules. What can I do? For functional enrichment analysis, I want to show you how to use combined gene list for further analysis. I analyze the code treatment with two traits, flower and height. I want to go further about influence details. So I have two ways based on the current result. The first way is to getting common gene set for target trait as DEGs. After we getting gene set, use functional enrichment result for check. Maybe we already have a large gene set for all DEGs and a smaller one for genes related with trait height and another gene set with trait flower. So we can get a smaller gene set with intersecting genes of all these three gene sets and using functional enrichment to check whether the pathway related to trait stress. Then we can select genes for expression design. The second way is collecting a common gene set for all genes which are interested in. Then using functional enrichment for further selection to get our final gene list. The difficulty of this approach is how to get gene lists based on functional enrichment results. But in BMKCloud, you can just combine two gene sets simply without distinct genes. I think it is very simple. Let's summarize here. Today, we talk about three topics. The first, we talk about analysis, save, and find. We're using Venn diagram as a case. You can save the results in BMKCloud or download the result on yourself. The second topic, we talk about WGCNA. I introduce the WGCNA meaning and the approach and the results. The last topic, we talk about functional enrichment. We shared two ways to get the final result. That's all. Thank you. Any questions?
Thanks for Johnson's. He explains the topic in great detail. I believe we have a good understanding of visualizing key pathways and WGCNA analysis with real data. Now we have received some questions about this. The first question is, for Venn diagram, can any part of the diagram be selected or just the intersect part? Ask for Johnson to answer for us.
Okay, I will show you. I did show the Venn diagram and selecting the intersect part, but for the whole gene set, but for the whole Venn diagram, you can select any part of this group. You can choose this part. Not only the intersect part, but also this part and not intersecting part can be chosen.
Okay. The second question is, does BMKCloud provide all results of WGCNA package or just the results you shared?
Oh, that's question. We shared about WGCNA results by some part of the results as an example. But if you download the zip file from BMKCloud, you will find that we provide all results from WGCNA packages. You can get them by downloading the whole zip on our platform, and you can check the results on your own. Thank you.
Okay, due to time constraints, we can't answer all questions, but we'll answer them by email after we have finished. As we conclude today's session, I want to express our gratitude for your participation. Look out for the follow-up email with the webinar summary and the promised recording, and make sure you check our social media page for future webinars. Thank you again. Have a nice day.